Now let us look at the cytology and the genetics of somatic embryogenesis. Okay. Now I told you, if you take carrot, okay, and then you grow this carrot in a test tube. You grow this carrot in a test tube. What will happen to this carrot? It will give rise to a what? It will give rise to callus. Yes. And from this callus, this medium is oxen rich. Okay. And then when you take this callus and grow in a non-oxen rich medium, what will you get? You will get these heart-shaped structures. And what do we call these heart-shaped structures? They are called as embryoids. Okay. Or in some books, meristemoids. Or non-zygotic, because there is no zygote stage, or non-zygotic embryos. Alright. Now these are embryos. Let us look at what is inside. What is inside this callus? Okay. And how do we know that this callus is going to give rise to embryoids? Then let us look at the genes, the genetic display of this callus. What is there? Okay. Does it signal? Does it give some information to a biotechnologist that it is going to give rise to an embryo-like structure, that is the embryoids. Okay. It has been seen that a callus, alright, a callus which is predestined, okay, which a callus which will give rise to an embryo. It is found that in this callus there are cells inside this callus. There are a group of cells. Okay, there are a group of cells. Now in these cells, it is found that if these cells are to give rise to embryos, then these cells will contain abundant cytoplasm. They will contain abundant cytoplasm. Then they will have irregular shaped mitochondria. Okay, mitochondria is like this. Okay, mitochondria is like that, but instead the shape of the mitochondria will be irregular. They will have abundant lipids. They will have free ribosomes. Then abundant endoplasmic reticulum. Okay, and beside that it will contain starch grains. And these starch grains will be found where? It will be found in the plastids. Okay, this cytology, okay, this information, abundant cytoplasm, irregular shaped mitochondria, lipids, free ribosome, all this indicate that this callus is predestined to form what? To form embryoids. Alright, then let us look at the genetic, okay, analysis of this callus. When we look at the genetic analysis of this callus, it is found that not all the DNA in this callus will be expressed. Not all the DNA in this callus will be active. Only a certain region shows intense DNA activity. Alright, it means that only the genes in those regions are very active, and they produces the proteins which are needed for what? They produces the proteins which are needed to produce non-zygotic embryos, that is the embryoids. Now these areas, alright, with abundant DNA activity, alright, they are known as the predestined cell. So what are PDC? Predestined cell. So these cells, they contain what? Abundant DNA. And this DNA activity is responsible for the production of proteins, alright? And it is these proteins which induces this callus to give rise to embryos, not any other somatic tissue, but embryos. Okay, now these are embryos. Okay, so these are the embryos. I want to make them into a seed so that I can store them. Okay, now we move to the next topic. These are the embryos. I want to convert them into a seed so that I can store them. So what will I do? I will take a beaker, and in this beaker I will add a matrix. What is this matrix? This matrix is mainly aluminum or sodium alginate matrix. Alright, then I take these embryos and then I put it here inside this matrix. After some time I take them out. Okay, and the matrix will form a covering, it will form an envelope, it will form a coat around these embryos. Now this behaves like what? It behaves like a seed coat. It is moisturizing or keeping moist whom? The embryo inside. So these are known as hydrogel. HYDRO. These are known as hydrogel because why? They feed the embryo inside by keeping it moist and by supplying nutrients to it. Okay. The moment these embryos are fully enveloped, fully coated by this sodium alginate mixture, they can be stored. They can be stored for 1 year, 2 years, or maybe 10 years. And when you need, you take the seed and then you grow in the soil. The moment you grow, this matrix will dissolve. This embryo will open out and then it will develop into the stem and the root system. Now these seeds are known as what? Now these are your seeds. They are known as the artificial seeds, or you call them as the synthetic seeds. So in traditional embryogenesis you get seeds through seeds. In somatic embryogenesis you also get seed, but after enveloping them in a matrix or a hydrogel, alright, and these seeds, they can be stored longer than the true seeds. They are known as what? They are known as artificial or synthetic seeds. The production of artificial and synthetic seed is one of the most important application of somatic embryogenesis, because you get seed which can be stored for a long time and they will remain viable whenever you want to grow them. That is the biggest application of somatic embryogenesis.